This is Cody McWilliams, and this is my light reaction and photosynthesis. My camera's down, so please excuse the phone quality. So starting over here, we have with what enters into the cycle through chloroplasts, and that would be water, photons of light, which are absorbed through the chlorophyll, and carbon dioxide. Now these go into the thylakoid, where they undergo electron transfer reactions from photosystems 2 to photosystems 1. Also inside the thylakoid is an electron transport chain. Now up here I just have some things talking about what's around the thylakoid and whatnot. So inside the chloroplast is the thylakoid space and the thylakoid membrane, which are surrounded by the stroma. And that is just kind of a little sandwich model of where everything's at. Anyway, back to the thylakoid. After those processes, oxygen is shown to exit through the chloroplast, and those processes are shown here. Starting with photosystem 2, light enters photosystem 2, higher energy electrons are transferred by plastoquinone, and then from there the cytochrome B6F complex catalyzes the transfer of these electrons from the plastoquinone to the plastocyanine. Forgive any mispronunciations. From then there is an embedded ATP synthase that uses the energy from an electrochemical proton gradient to produce ATP. And down here we talk about, because the mobile electron carrier removes an electron from the reaction center, an oxygen evolving complex, also known as the water splitting complex, uses manganese atoms that hold water molecules in order to extract these electrons, and this is what supplies photosystems 2 with electrons. Now photosystem 2 produces ATP. The process continues with photosystem 1, and here ferredoxin catalyzes the reaction for NADP plus with hydrogen plus to produce NADPH. Ferredoxin NADP plus reductase is the last enzyme used in photosystem 1. The NADPH that it produces is going to be used later in the Calvin cycle. And now entering the Calvin cycle, I've broken it down into 3 steps. It's products and then the net products. So starting with step 1, it begins with ribulase bisophosphate, or RUBP, which is consumed by the addition of a carbon dioxide. This is done by the enzyme Robusco, which attaches the carbon to ribulase 1,5-bisphosphate. This step ends in the production of a total of 6 3-phosphoglycerates. 2 of these 3-phosphoglycerates come from each CO2 molecule that are initially entered. From there we go to step 2. Just as following the first step, ATP and NADPH are consumed. This produces glyceraldehyde 3-phosphate, or G3P, and a total of 6 of these G3Ps are produced from this step. Moving on to step 3, of these G3Ps produced from the previous step, some are used in order to regenerate ribulose 1,5-bisphosphate, and only one of those is used of the original 6. So the remaining 5 of 6 G3Ps leave through the chloroplast stroma and enter into the cytosol. These ribulose 1,5-bisphosphates are catalyzed by an enzyme called carboxylase. And I'm sorry, I meant to switch up those 2 bullet points for order, as you can see. Moving down to products. The carbohydrate products of the Calvin cycle are 3 carbon sugar phosphate molecules. These are G3Ps, and glucose is also the main product of the Calvin cycle. And then for net products, for every 3 carbon dioxide, the cycle produces 1 G3P molecule, and 9 ATP and 6 NADPH molecules are used. Again, those NADPH molecules come from the light reaction and photosynthesis.